Our very special guest today is Dr. Catherine Xu, who I met, 16 years ago, something absurd like that, back in the days of high school philosophy club and science Olympiad. We were the co-founders of the philosophy club. I am proud to say. I should put that on my CV. But after all that fun, Catherine went to Harvard, got her bachelor's in chemical and
physical biology. They just made up that
way
major.
Too many subjects in there. Then stayed a year to work as an associate editor at Harvard's magazine, and then moved on to do a PhD in genomic science at the University of Washington, where she studied, as you see here on the screen, the evolutionary dynamics of the flu. And then, if that weren't enough, went on to do, or is currently doing, a postdoc at Stanford or Sanford or Sandford, whichever one you prefer to use, where she studies the human microbiome and a little bit of COVID action now, I believe, as well. So I'm very happy to introduce our guest, Dr. Catherine Xu. Go ahead.
Thank you so much, Paulo, for that very kind introduction. It's really fun to kind of reconnect over all of this science stuff now. And thank you all for inviting me here today. I'm really excited to share with you some of my research on influenza evolution. So before I get started, I just wanted to mention, although I'm currently at Stanford, everything I'll be sharing today is from my PhD, which was in Jesse Bloom's lab at the Fred Hutchinson Cancer Research Center and University of Washington in Seattle. And second, I just wanted to mention, please feel free to interrupt with questions. Don't be shy about speaking up. I think especially in this Zoom era, it'll probably be more fun all around if we just kind of make this more of a kind of back and forth, and I have planned timing accordingly. Okay, so let's get started. So influenza evolution has had a really profound impact on human history. Our modern influenza era really started a little over a century ago with the 1918 to 1919 influenza pandemic. Now, this pandemic was the single most devastating pandemic in human history, killing anywhere from 50 to 100 million people. Now, this pandemic was the product of a zoonotic transmission. The vast majority of influenza's genetic diversity is within its main host, I shouldn't say species because it's a very wide group, it's birds, specifically waterfowl. So most influenza genetic diversity is really within avian influenzas, but occasionally, as in the case of the 1918 pandemic, a virus will acquire the mutations necessary to adapt to infecting mammals like humans instead. So this pandemic, as I mentioned, was incredibly devastating. And even after the pandemic, this H1N1 subtype virus that was introduced within the pandemic continued circulating for another 40 or so years. And that went on until 1957, which was the next influenza pandemic. Now, this flu pandemic was caused had a different origin than the 1918 pandemic. Specifically, influenza viruses are extraordinarily capable of gene reassortment. Their genes, they have 8 different gene segments, which you can think of basically as separate chromosomes. And it's possible for these flu viruses to mix and match these gene segments between different viruses. And that's what happened here. This H1N1 virus introduced in 1918 was able to mix and match its genes with an avian influenza virus, creating a new reassortant virus that carried some of this H1N1 skeleton, but had different antigenic proteins. Specifically, it was of this H2N2 subtype, and those H's and N's are referring to hemagglutinin and neuraminidase, which are the two main surface proteins for flu and the main antigenic determinants. So this H2N2 virus circulated for about the next 10 years seasonally, and that went on until our next flu pandemic, which was in 1968. So this pandemic was again the product of a gene reassortment event, which produced the H3N2 flu strain that actually continues to circulate seasonally today. It's one of 2 subtypes responsible for our yearly influenza epidemics. Now, although this green bar may look very stable on this slide, the fact is that the virus has actually continued changing throughout those multiple decades. In particular, it's undergone this really interesting and important process of antigenic drift, whereby the virus is constantly acquiring new mutations that allow it to infect and reinfect previously infected individuals. And so this is a major reason why we have to update the annual flu vaccine every couple of years. Okay, so this all makes it seem like the history of flu is pretty straightforward. We've just got one dominant flu strain at each time. That's not quite the case though. So let's go back to this H1N1 strain that was introduced by the 1918 pandemic. It actually, after it disappeared in 1957, it actually reemerged in 1977 to cause a relatively minor flu pandemic. And I say relatively minor because it only impacted a relatively small proportion of the population, namely individuals who were younger, children and young adults who had not yet been exposed to the virus when it circulated in the 1950s. Now, the origins of this 1977 pandemic are still a little bit of a mystery, but molecular dating actually shows that the virus did not acquire any additional mutations between the mid-1950s when it disappeared and 1977 when it reemerged. And so the leading hypotheses today are that this pandemic was caused either by a laboratory escape or by a vaccine trial gone wrong. And my impression is that the current speculation leans a little bit more heavily towards the second of those possibilities. But again, we actually just don't know. This 1977 pandemic reintroduced this H1N1 virus into circulation, and it continued to co-circulate with H3N2 for the next several decades until 2009, which was our most recent influenza pandemic. So this pandemic, which I think many of you may remember, was kind of nicknamed the swine flu. And that's because it was, again, a reassortant virus. This time, the virus had picked up some genes from a strain of influenza that had actually been circulating in pigs since 1918. So it was actually introduced into swine in parallel to when it was introduced into humans in 1918, and it actually continues to circulate today. Anyhow, this 2009 virus had picked up some of those genes from the swine lineage, and that again changed its antigenic properties. Although it was still of this H1N1 subtype, we call this subtype pandemic H1N1 just because it's genetically actually quite divergent from the previously circulating H1N1 subtype. Okay, so I hope that all of that just kind of gives you a sort of 20,000-foot view into why influenza evolution is important. And I hope this drives home the point that influenza viruses are capable of evolving extremely rapidly with many important consequences for human health. Now, over the past few decades, influenza evolution has been pretty extensively studied, and it's really become this poster child for understanding rapid positive evolution. So, for instance, here I'm showing you a phylogenetic tree depicting influenza sequences of this H3N2 subtype from over the past 4 decades. And what we see is that this tree has this unusual kind of ladder-like structure. People call it ladder-like or spindly or unbalanced. What all of those terms mean is just that of the two descendant lineages from any particular node, one lineage tends to leave a disproportionate number of the offspring. And this shape of the tree is a strong signal that there's positive selection going on that's favoring certain lineages to take over the population and replace other lineages. To get a sense for what that evolution might be, we can overlay some of the antigenic characteristics of these viruses. So each of the colors here represents a group of viruses that are recognized very similarly by the immune system. Different colors are recognized, have accumulated enough differences that the immune system recognizes them slightly differently. It doesn't mean that we've lost all immune memory, but it means things have changed enough that we may need to update the flu vaccine and people may be reinfected. And so you can see that at any given point in time, you tend to have one or two of these antigenic clusters circulating, but over time, these antigenic clusters replace one another. So again, this really vividly shows us that flu viruses are constantly evolving in order to escape host immune defenses. Now, although these sorts of phylogenetic analyses have taught us a lot about flu evolution, I would argue that there's still a lot that they overlook. The reason is because each of the tips on this phylogenetic tree that I'm showing you represents the infection of a single individual. And that's summarized in the form of one consensus sequence, which kind of represents the average genetic identity of all of the viruses within that population. Now, a given person's infection typically actually has billions, trillions, or even more viruses. It's actually very difficult to count. And that enormous viral population can harbor pretty significant genetic diversity. More recently, using high throughput deep sequencing, we can start to measure some of this fine scale genetic diversity that may be present within these influenza populations. In particular, we can identify variable sites that we may not detect using consensus sequencing alone, but that still may tell us about a lot about the virus's evolution. And so this is what I'm going to tell you about today. Throughout my PhD, I use deep sequencing as kind of a microscope for viral evolution. We can use high throughput sequencing to really zoom in on influenza infections to ask the question of how influenza viruses evolve at very small scales of space and time. So I'll tell you broadly two different stories from my PhD work that touch on this topic. Okay, the first story starts about 15 years ago when various clinical flu labs started noticing that when they took clinical flu strains and grew them in cell culture, a certain mutation would arise again and again. Now, for this part of the talk, I'll refer to the wild type virus in blue. And so it has an aspartic acid at site 151 of the neuraminidase protein, which is one of those two proteins I told you about earlier. It's on the viral surface and is responsible for viral exit from host cells. Now, this red, I'll refer to this mutant virus in red. So it has a single nucleotide mutation that changes the identity of the amino acid at site 151. And as we'll see, that actually has some pretty important consequences for these mutant viruses. Now, the prevailing thought about this D151G mutation was that it was probably a lab adaptation. And in fact, it is very common for influenza to adapt both in laboratory settings and actually within vaccine development, which tends to happen in eggs. Now, there were a couple things that suggested to us, though, that this lab adaptation wasn't really the whole story. For one, although many groups had observed that this wild type and mutant virus coexisted in cell culture, very few groups had reported cases in which this mutant virus actually took over the population. Now, if it were truly an adaptive mutation, you would really expect it to outcompete this wild type virus, but that wasn't what we were seeing here. Instead, it seemed like the two viruses would persist at intermediate frequencies. Now, another puzzling observation was that a different group did a biochemical study of this D151G mutation. And what they found was that this mutation actually destroys the catalytic activity of the protein that it's in, which again is responsible for helping newly formed viruses exit host cells. So effectively, this D151G mutation seems to be a loss of function mutation. So it wasn't really clear to us why it would be tolerated at all, let alone be some sort of adaptive mutation. So to try to understand this a little bit better, we decided to compare the growth of the wild type and the mutant viruses in the lab. And so here I'm showing you a growth curve with both the wild type and mutant viruses. You can see that the wild type virus in blue grows pretty well, and the mutant virus actually grows quite poorly. The titers that you see on the y-axis are on a log scale. So this was in line with all of that biochemical evidence, right, which suggested that this mutant virus has a loss of function mutation that makes it very difficult for this virus to replicate. But it didn't really answer why, in that case, this mutation would be arising in the first place. So to try to answer this, we went back to that observation that the wild type and the mutant viruses often coexist in cell culture. We assembled a 50-50 mixture of the wild type and the mutant viruses, and then we infected them onto cells in cell culture to again do this kind of growth curve. And this is what we found, to our surprise. So we saw that this mixture of the wild type and mutant viruses actually outgrew either population of just one virus alone. So it really suggested to us that these two viruses were able to cooperate with each other. They were able to grow better together than they could apart. Now, here I want to take a brief detour to talk about this general concept of RNA virus cooperation. So RNA viruses like influenza or SARS-CoV-2, they mutate very rapidly. And so that means that anytime you have just a viral population, it harbors significant genetic diversity. In fact, some people have called this a kind of quasi-species effect, meaning that you always have this cloud of genotypes interlinked through mutation. Now, there have been various theories and some evidence suggesting that viruses within a population can actually cooperate with one another to improve the overall population fitness. Until now, though, there hadn't really been specific molecular examples of this kind of cooperative interaction, but that's what we thought we had identified within this influenza system. Now, if the viruses are cooperating with one another, there are a couple predictions that you would make. You would expect that if you take this mixed population of wild type and mutant viruses and then infect it and then grow it for some time in cell culture, you would expect that if both viruses are really contributing to the fitness population, to the fitness of the overall population, then both viral variants would be maintained. That is to say, you wouldn't have one virus driving the other one extinct. You would also expect that if you start off with pure populations of either the wild type or the mutant viral variant, then the opposite variant will arise through de novo mutation, and then it will increase in frequency so that this population will still eventually converge to this mixed population state. So we tried this experiment. We started off with these wild type and pure populations of the wild type and mutant variants, as well as a 50-50 mixture of the two. We grew them for 5 passages in cell culture, and then we used deep sequencing to assess the proportion of the 2 viral variants. And this is what we found. So here's just a small example of our data to orient you to our results. So here I'm showing you a case where we started off with a pure population of the mutant viruses in red. And then this wild type virus in blue actually starts making up a substantial proportion of the population by the second passage in cell culture. And then over time, it eventually kind of reaches this equilibrium of about 50-50. Now, if we start with that 50-50 mixture, we see that that's actually maintained quite stably throughout these multiple cycles of growth in cell culture. And conversely, if we start off with a pure population of the wild type viruses, we see that the mutant viruses do eventually arise as well. Now, we tend to see that this mutant virus arises more slowly within a wild type population compared to the wild type population emerging in the mutant population. But that makes a lot of sense to us given the relative growth rates of these 2 viral variants in isolation. Now, these results are extremely consistent across 3 biological replicates. And in fact, I actually want to draw your attention to this N151 variant in green here. So this N151 variant is very interesting. It has not been biochemically characterized like this G151 variant has, but it in many ways seems to behave very similarly. That is to say, in clinical labs, it often arises in concert with this D151 variant. It doesn't seem to take over. And we did some additional studies of this N151 variant to suggest that it behaves very similarly to G151. That is to say, it grows very poorly in isolation, but it grows well with the wild type variant. So we think this is another example of a potential cooperative interaction. Okay, so you might be wondering at this point, how does cooperation happen? So we've done some work on this. A lot of it gets very virusy, for lack of a better term. So I will just try to give a general overview of what we think is going on. So viruses have this very unusual property that if you have 2 viruses of distinct genotypes that happen to co-infect the same host cell, then both distinct viral genomes will be deposited into the host cell, both protein variants will be produced. And then the progeny viruses are going to package on their cell, well, not their cell, just the virion surface. They're going to have both protein variants, even though they only package one copy of the viral genome. So that means that they're basically phenotypically mixed, even though they genotypically only carry a single copy of the viral genome. So this is something that we call genetic complementation for viruses, and we think that it may be contributing to the cooperative interaction that we observe. So one line of evidence supporting this is the fact that we see that the dynamics of viral cooperation appear to depend on the virus, on the infecting multiplicity of infection, or MOI. So MOI is basically just the density of viruses in cell culture. At low viral densities, you expect there to be less co-infection. At high densities, you expect more co-infection. So at an MOI of 0.02, meaning that on average 2% of the cells are expected to be infected with at least one virus, we see that this mixture of the wild type and the mutant populations, this mixed population does not actually outgrow the wild type population, at least at the early point of this growth curve. Now, it does eventually actually outgrow the wild type population, but we should remember that at this point, multiple cycles of viral replication have taken place within this single passage in cell culture. And so the effective viral density by the end of this experiment is very high. And so we think that, cooperation is only really kicking in at this higher MOI. In contrast, if we start these populations at a very high MOI of about 0.5, then we see that indeed this mixed population does outgrow the wild type population. So again, we also have some more functional evidence that kind of lines up with this, but I'm happy to get into that if people are curious. But this is just kind of the general idea. To say a little bit more about the specific mechanism of interaction, we suspect that this mutant and wild type virus may have kind of complementary functions, at least at the protein level. So the wild type function of this neuraminidase protein, as I've mentioned, is to cleave newly formed viruses off of host cells so that they're able to go on to infect new cells. Now, from this biochemical study, other groups have shown that this mutant virus, this G151 virus, retains the binding activity, but it doesn't have that cleaving enzymatic activity. And so that's quite interesting because flu actually uses the same receptors to both enter and exit host cells. And it seems like this mutant virus probably has enhanced cell entry activity. And so we think that in some cases, having a mixture of this wild type and mutant activity can actually be beneficial to viruses, at least in a cell culture setting. So for example, it's been shown that recent flu strains, when grown in cell culture, have kind of relatively inefficient viral entry properties. Now, of course, they are quite capable of entering the host cells that are found in the human lung. This is something of a cell culture artifact, just because the cells that we use to grow viruses are not as complex as, of course, the human lung. Now, although empirically, these recent flu strains seem to bind weakly to cells in cell culture, they still exit quite efficiently. Now, we think that for the mutant virus, we suspect that it has very enhanced cell entry activity, just because it has that kind of supplementary binding activity of this protein that's usually involved in viral exit. Now, the problem for these viruses are that they then have no very good mechanism for viral exit. Sometimes it seems like they'll probably just drift off of these receptors, which is why we get any growth at all. But this is not a very viable strategy for this virus in isolation. Now, when these two viruses are present in the same population, what we think is going on is that there's genetic complementation, so that you have progeny viruses that carry both viral variants. And so we think that these progeny viruses, just due to this kind of unusual cell culture situation, kind of have the best of both worlds. They have very efficient viral exit because they still have some of that activity of the typical wild type neuraminidase, but they also have enhanced viral entry because they have that binding activity of the mutant protein. So again, if folks are interested in more kind of virological details about this, I'm happy to go into our other lines of evidence, but this is just kind of a high-level overview of what we think is going on. To wrap up this part of the talk, I want to just touch on another question, which is whether cooperation occurs within natural clinical infections. Now, I told you at the start that this mixture of viral variants was first identified when clinical influenza samples were grown in cell culture. And so a big question for us was whether this mixture of viral variants was present within natural infections or whether it was something that arose most particularly within cell culture. To answer this question, we worked with collaborators at Washington State Public Health Labs and UW Virology to identify and then directly deep sequence 9 clinical samples. And so these were samples that we knew had formed mixed populations after being grown in cell culture. And just to provide some context, that's often what's done if people are trying to do different assays to measure the antigenic properties of these viruses. So we were able to find the original clinical samples. We could sequence them prior to passaging. And when we did that, what we found was that that the mutant virus was undetectable within these actual clinical samples. And so that suggested to us that this cooperative phenotype, as I've kind of been suggesting, as I've described it, is something that is kind of particular to cell culture conditions. Now, there are a couple reasons why this might be the case. Of course, there are many, many differences between actual human lungs and the simplified systems that we use in the lab. But we think the most relevant differences here might have to do, one, with that viral density question, right? So we grow viruses to extremely high titers or viral densities in cell culture. That is kind of the whole point of cell culture. And I think that provides a kind of unusually good setting for these viruses to interact with one another. The second major feature is that within natural human infections, the mucus lining of the respiratory tract plays a major role in preventing viral infection. And the wild type activity of that neuraminidase protein is actually something that typically helps the virus get through that mucus layer. And so it seems like this mutant virus may suffer an even bigger disadvantage within natural human infections compared to cell culture for that reason as well. Okay, so to summarize this part of the talk, we used deep sequencing to zoom in on what appeared to be a case of simple laboratory adaptation, and instead we found something quite a bit more complicated, which was this kind of odd cooperative interaction that seems to have arisen repeatedly in many, many labs in hundreds of cases around the world. So it's a very robust and repeatable example of viral cooperation. That said, this viral cooperation does seem to be constrained to clinical infections, constrained to cell culture settings rather than natural clinical infections, which is probably something we should all be grateful for. Okay, so this kind of wraps up the first half of the talk. I just want to pause at this point in case anyone has any questions at this point. All right. Well, if everyone feels satisfied, I can continue. Great. Okay. So in the second half of my talk, I'm really going to build on this kind of last result that I showed you about how cooperation doesn't seem to arise within clinical viral infections. So, you know, this really got me thinking about the question of, you know, what is going on within clinical viral infections? Clearly, cooperation is not something that seems to be happening, but clinical infections do provide opportunities for viruses to accumulate genetic diversity. And so I wanted to try to answer the question of how influenza viruses evolve within hosts over the course of an infection. And I think this is an interesting question to think about from a basic evolutionary point of view. And just to kind of help you understand the context, I want to give you a couple key parameters for thinking about influences within host evolution. So flu infections typically start with a very small founding population of viruses. Various sequencing studies have suggested that the effective genetic bottleneck for flu probably is about 1 to 2 virions. So that does not necessarily mean that, that you are only being exposed to one virus. What it means is that empirically, the start of an infection is quite stochastic, and it seems like when an infection actually does successfully get underway, it's usually just one or at most a few viruses that are responsible for leaving all of the offspring. So we start from this very small founding population that rapidly and exponentially expands and is able to generate genetic diversity. The typical course of a flu infection lasts about 5 to 7 days with a peak viral load around day 2. We've actually spent considerable time trying to figure out how many viruses on average are in a person at that point, but it happens to be something that's incredibly hard to figure out. So I don't even have a good order of magnitude for you there, but it's a lot of viruses. Okay, so the mutation rate of these RNA viruses is quite high. And so 1 in 10 to 1 in 100 viruses, newly formed viruses are going to have at least one mutation in the genome relative to the parent virus. And so within a population size, that at least census population size, as best we can guess, is like trillions at least, that is a lot of genetic diversity. Every potential single mutation is definitely being generated in every infection. So we are not mutation limited here. So I really wanted to understand in the rest of my PhD how this genetic diversity that emerges within every new flu infection can actually guide the virus's overall evolution. And I think this is a really important question simply because all of the viral variants that all of flu's genetic diversity ultimately has to start with individual mutations that arise within single infected individuals. So only a very small number of these mutant viruses that arise within an individual will go on to transmit from individual to individual, and an even smaller proportion goes on to spread or even take over on a global scale. So after wrapping up that cooperation project, I decided, okay, I want to try to understand how viruses are evolving within individual patient infections and how that fits into this broader picture of flu's global evolution. So I got very lucky at the outset here. Starting off with this project, we didn't really know where to start. At the time, Jesse's lab was mostly a basic science lab. Now we have very many clinical collaborators on various different dimensions, but that wasn't quite the case yet at the time. And in fact, I have this wonderful email from my advisor, wow, back in 2015, talking about how he ran into some clinicians at the Fred Hutch while getting coffee, and they had sort of realized that these clinicians had collected a really extraordinary set of flu samples that might shed light on the virus's evolution. And so this was the start of a really rewarding and wonderful collaboration, the results of which I will tell you here. Okay, so as I mentioned, typical flu infections last about 5 to 7 days. Now, this feels like a terribly long time, of course, while you're sick, but from a scientific standpoint, this actually doesn't provide us a whole lot of time for understanding the virus's evolutionary dynamics, that is to say, the change over time. The vast majority of studies of influenza's genetic diversity up to this point had just been cross-sectional, which is to say people deep sequenced a single sample of the virus collected from during the course of an infection. Now, this is still very important, of course, but the static snapshot of the virus's genetic diversity provides us only limited information about what these mutations are doing and why they're arising. In our study, working with these fantastic collaborators, we studied this really unusual cohort of 4 immunocompromised individuals who had extremely lengthy flu infections lasting, in this case, 2 to 3 months. So in the course of regular clinical care, our collaborators had collected viral samples from approximately weekly throughout the course of these infections. And by deep sequencing these viral samples, we hope to get a sense for how and why these viruses were changing over time. So here's just a small snapshot of what we found. So here I'm focusing in on a single patient, patient W, and all of the mutations I'll be telling you about in this part of the talk are in hemagglutinin. And so that's the H of the H3N2, and it's the main antigenic determinant for the protein, for the virus. Okay, so within this one gene and the viruses within this one patient, we actually saw substantial evolution. In some cases, like here with this A138S mutation, we saw a virus kind of increasing in frequency, but then declining in frequency by the end of the infection. In other cases, we saw this F193Y mutation arising very early, kind of seesawing in frequency, and then actually going on to take over the viral population. Now, this was really fascinating to us. We hadn't really known what to expect when we started this work, but this all suggested to us that viruses were capable of evolving extremely rapidly over the course of these multi-week individual patient infections. Now, something you might be wondering here is that there's a lot of kind of evolutionary change going on, but a lot of it seems rather contradictory. You have mutations that increase in frequency and then go down in frequency again. So what's that all about? Well, to understand that, I think you need to understand a particular feature of the flu genome. So flu has 8 distinct gene segments, and each of these segments are capable of reassorting with other similar segments. You can think of them kind of as chromosomes, but there's no equivalent of homologous recombination for flu. What that means from an evolutionary point of view is that each gene evolves clonally. There's no recombination. And that has really important evolutionary consequences. Now, typically, when we think about an adaptive mutation, we would expect an adaptive mutation that arises to eventually take over the population. However, in very large clonally evolving populations, you can sometimes get multiple adaptive mutations arising simultaneously, like the orange, green, and purple here. And so these mutations will actually compete with one another and prevent any single mutation from taking over the viral population. Now, this is a phenomenon known as clonal interference. It's been extensively observed within experimentally evolving populations like bacteria in the lab, and we thought that it might be affecting the trajectories of mutations within these viruses as well. So to try to understand this a little bit better, we focused in on 4 mutations within the hemagglutinin gene of this patient that I showed you earlier. And these 4 mutations happen to be located close enough to one another on the gene that we could actually get individual sequencing reads that would span all 4 mutational sites. So that meant that we could then assess, we could then phase the mutations, right? We could figure out what combination of mutations was present in different viruses over time. And so that's what I'm showing you here. Here, each of the colors is a different combination of identities at those 4 genetic sites. This gray lineage here is the wild type lineage, which I'm denoting with 0 at each of those 4 sites. And you can see that over the course of the infection, the wild type lineage actually winds up disappearing. By the end, all of the viruses have at least 1 mutation in the hemagglutinin gene. Okay, so we see multiple single mutant lineages arising pretty early within the infection. And in fact, this orange lineage that takes over eventually is the one that has this mutation. And so it eventually takes over the population. I should, of course, note that we're only looking at the mutations at these 4 sites. And so the viruses that eventually fixed here may actually also have carried additional mutations in different viral genes as well. Okay, so we see various single mutant lineages arising. We also see a double mutant lineage that reaches a pretty high frequency. And so that's the double, that's a virus that carries the mutation at site 223 and 225. And so I think you can see, just based on the very similar trajectories of these two variants, that it looks like they're linked, right? And that is indeed the case. Now, what was kind of surprising, though, was we also saw various triple mutant lineages. Okay, so viruses that had 3 of these 4 different mutations that we observed. And so this was just really amazing to us for a couple of different reasons. One is that we see here that these mutations are really arising in different, are arising and reaching high frequencies in different combinations, which is pretty extraordinary. This is a very, evolution here is clearly not mutation limited, right? Just like I alluded to earlier. Now, the other interesting point here was that we can actually identify, by reconstructing the kind of paths through which these mutations arose, we can actually kind of say, we can infer that certain mutations like this A138S mutation here at the first site, that mutation must have arisen at least twice independently. So it must have arisen on this wild type background to form the single mutant lineage, and it must have arisen independently on this double mutant background to form the triple mutant lineage. So again, we know this just because there's no mechanism for homologous recombination within genes for influenza. But again, I think this really drives home the immense selective pressure that these viruses are under within hosts to come up with new evolutionary solutions. Okay, so far I've really focused on evolution within just one individual, but how does evolution compare between the different patients in our study? So to our surprise, we found that some of these mutations that we observe arising, we saw that many mutations actually arose independently in multiple patients in our study. So for instance, this A138S mutation arises within patient W, as I've told you, but it also actually winds up taking over the population in patient Z. If we look more broadly across the hemagglutinin gene, we see many sites of which this is the case. In some cases, the same mutation actually reaches high frequencies in 3 of the 4 patients in our study. Now, this is a lot more similarity than you would expect purely due to chance, and it suggests that really similar selective forces are driving evolution within these different patient contexts. And we can get a sense for why that might be if we plot the locations of these recurrent mutations on the crystal structure of the hemagglutinin protein. Most of these recurrent mutations cluster within a particular region of the protein, which is responsible for viral binding to receptors on the host cell. And so this region is also a major target for human antibodies. That leads us to think that selection to escape the immune system is driving a lot of influenza's evolution within hosts, just like it's driving evolution on this broader between host scale. Now, there are some exceptions. Keep an eye on site 427 here, which I'll talk about more in a second. It's somewhere different than all of the other mutations. Okay, so I started off by telling you that I was interested in understanding how influenza's evolution within hosts compares to its evolution on a global scale. So here again are the mutations that we observed recurrently in multiple patients in our study within the hemagglutinin gene. Now, we wanted to know whether any of these mutations were found in the global flu population as well. And the answer is yes. So for instance, here, this A138S mutation is found in 2 of the 4 patients in our study. It also reaches relatively high frequencies in the global flu population. So you can see that as of about 2016, it made up about a quarter of circulating flu strains of this subtype. Looking more broadly across these sites of recurrent mutation, we see some cases in which the mutation that we observe within the patients in our study actually goes on to reach fixation on a global scale. Now, there are some exceptions. So here's site 427 again. This is a weird one because we see this mutation reaching very high frequencies in 3 of the 4 patients in our study. It is never observed in the consensus sequence of a flu infection in the global population. So this one is quite a bit of a mystery to us. Here we think maybe this mutation represents some sort of trade-off between viral replication within hosts and transmission between hosts. That's something that's been theorized, but there aren't that many concrete examples of mutations that bring that trade-off into effect. So yeah, hopefully other folks down the line can do more tests to try to figure that out. All right, so here I'm showing you just a subset of the mutations that we identified, but we did more systematically compare the mutations that we identified within hosts compared to on a global scale for each of the different influenza genes. And what we found was that sites of within host and global evolution showed statistically significant overlap within the hemagglutinin gene, but not in the rest of the flu genome. And that does make sense to us just because this hemagglutinin gene is still the main antigenic determinant for these flu viruses. Okay, so I just want to wrap up this part of the talk by commenting a little bit on the relevance of our work today, given the context of the SARS-CoV-2 pandemic. So one thing that's been really fascinating for me to see is that there have been various groups doing studies, similar studies, tracking the evolutionary dynamics of SARS-CoV-2 within long-term infections, again, usually within immunocompromised patients. And as these two studies show, at least in some cases, not all, but in some cases, people are observing pretty dramatic evolution over the course of multi-week infections. In some cases, the mutations that arise during these infections can actually help confer escape, for instance, from antibody cocktails that are used to treat the virus. And so I think that really shows the importance of thinking about evolution on this within host scale. And in particular, in the case of influenza, we don't really think that immunocompromised patients are a major vehicle of flu evolution. But there's been some speculation that this may actually be happening for SARS-CoV-2. In particular, the B117 variant lineage, which was first identified within the UK. There's a lot of speculation that it may have arisen within a long-term patient infection. The reason for this, there's not direct evidence, but the reason for the speculation is because the UK has actually done very extensive genome sequencing of SARS-CoV-2 infections, much more extensive sequencing than the US has done. But this B117 lineage, the viruses all carry this cluster of 5 or 6 mutations that just were not identified within any other sequenced viruses. And so the speculation is that all of those variants may have arisen, simultaneously within a single person's infection, which is why they otherwise had not been detected. So again, I should emphasize this is speculation. I think it's very difficult to definitively establish the origin of any one of these lineages, but I do think it's important to think about the context of these long-term chronic infections, which often offer more sustained periods for viruses to evolve. In other work that I've done, which I didn't have time to talk about here, we've looked at evolution within acute flu infections, the ones that just last 5 to 7 days. And here, a major force that kind of slows down evolution is the fact that the virus undergoes a really severe genetic bottleneck every couple days. It expands, accumulates genetic diversity, and then loses practically all of it. So that's immense genetic drift. And we think that is partly why within these longer chronic infections, these viruses are able to evolve so much more rapidly. Okay, all right, so to sum up this part of the talk, we see that influenza viruses can evolve extremely rapidly within individual patient infections. We see similar mutations being selected multiple times within a single infection in different patients in our study and also on the global scale. So again, I think this really points to the power of deep sequencing to really help us understand flu's evolution. Okay, so I want to take just a moment here to comment on what I'm working on now, as Pablo alluded to. So at Stanford, I've shifted a little bit to studying adaptation and dispersal within the human microbiome. So again, evolutionary dynamics, just in the context of a multi-species, very complex community. So effectively, what I'm interested in studying is how these bacteria within the microbiome are adapting over time and how they're transmitting between individuals simply because, we acquire all of our microbes from the people and places around us. And so something I've been working on is to run a human subject study. This is the first time I've done this. And it is a study of household transmission in the human microbiome. So we have recruited actually now more than 20 pairs of healthy cohabiting individuals, and they have undergone a longitudinal sampling protocol. So they're collecting microbiome samples over the course of 2 months, with more frequent daily sampling in the middle of the study. And that's around when one of these 2 individuals takes an experimental course of the antibiotic ciprofloxacin. And this antibiotic is used as a microbiome perturbation to try to understand the effects that transmission might have on subsequent microbiome recovery. So I think you can see a lot of similar themes to the work that I've shown you from influenza. I'm still working with a lot of deep sequencing data, longitudinal sampling, just in basically just with poop instead of snot. So on that note, I'll wrap up. I want to thank my PhD advisor, Jesse Bloom, as well as our many collaborators, especially our clinical collaborators at the Seattle Cancer Care Alliance, Washington State Public Health Labs, and Mutant Virology, who made all of this work possible. And I've also been really fortunate to be supported by various sources of fellowship funding. And I'm really grateful for the independence and intellectual freedom that those fellowships have given me. All right, so with that, I'm happy to take any other questions that you all may have. And thanks for your attention.
That was an amazing talk. I really enjoyed it. I am curious about the patients that were immunocompromised. Was their overall immune capability monitored? And how would this, if they have spikes in it, how would this relate? Could this relate to the modulating of the diversity of the population of influenza viruses?
Yeah, that is a great question. And I think it really gets at this fundamental paradox. I told you that these folks are immunocompromised, which is why they have such long infections. And yeah, what we're seeing seems to be pretty clear antigenic evolution. So evolution to escape the immune system. So these 4 patients are all bone marrow transplant recipients. They got flu infections following the engraftment of the bone marrow transplant. We do have various immunological metrics, just kind of like the number of lymphocytes over time. They're actually quite noisy. I mean, it's a very complex patient context because you have this very complicated transplant taking place alongside, this very complicated flu infection, and also actually simultaneous other respiratory virus infections too, which I didn't mention. So to your question about whether that may be influencing some of the dynamics, I think it's a little harder for us to say that on the immune side, simply because we don't have a great sense of how the immunological measures correlate to immune selection. But I can say that in some cases, patient treatment has had a really clear influence on flu evolution. So I didn't show this example, unfortunately, but these patients did undergo a very prolonged treatments in some cases with the antiviral drug oseltamivir or Tamiflu. And so in some cases, I mean, we also see extensive evolution of these antiviral resistance mutations. And in some cases, they actually emerge just very quickly following the initiation of this antiviral treatment. So we can see some of these, how some of these host or kind of clinical treatment factors may influence the viral evolution.
Thank you so much. My follow-up would have been about treatment, so you already answered that.
Great.
Thank you.
Wonderful. Any other questions?
Yeah, Jensen. I was really intrigued by the part of the study involving the immunocompromised patients. And since these variants can persist for longer periods of time in them, does that lead to reduced virulence over time?
Yeah, that is a really good question. We don't know too much about the virulence of those particular viruses, and I think, you know, the jury is kind of out on whether the sustained evolution would increase or decrease virulence, and also an additional factor like transmissibility, right? There is, I think, you know, a lot of theories suggesting that, okay, maybe there's this trade-off between virulence and transmissibility. And so as the viruses evolve long-term within a person, they may just be kind of less able to then infect other people. But it's unfortunately incredibly complicated to assess just because of the complex patient context as well. But I think that's a great question.
Could I actually follow up? So what I was also wondering was, how long has the use of immunosuppressant drugs been around? And related to that, has that led to increased variant production in human populations?
That's a really good question. I don't know much about the history of immunosuppressant drugs. I think overall, there has been some speculation for various viruses that immunocompromised individuals may wind up being kind of reservoirs of viral infection and generation of new viral variants. I think it's, as you can see with this B117 lineage, it's very difficult though to kind of pinpoint the exact origin of these variants. And so although I think it's very interesting to think about, we just haven't yet kind of figured out those direct links. But in the case of, say, SARS-CoV-2, you know, there's been such intensive attention to the virus that we may start to learn more about those kinds of links between immunosuppression and viral evolution. Good question. Great. Anyone else? We're right about at 2 o'clock.
Well, could I ask another question?
Go ahead.
I'm curious about the use of the term cooperation, and that I've never heard it used in this context before, and was sort of very intrigued by that. And is that really the best terminology, or I mean, I don't know, just.
Yeah, I think we've discussed this with a lot of folks previously too. A lot of evolutionary biologists define cooperation as kind of altruism, which is something that one organism is doing that imposes a cost to itself, but then a benefit to to another organism. And so in this case, we don't observe an obvious cost, at least to the wild type variant, right? The mutant variant, you know, might fit that definition a little bit better. So I think in that instance, our case doesn't really line up with the typical evolutionary terminology. You know, we could also call it, I think, more of a mutualism. We were kind of, alluding more to this lineage of work on general RNA virus cooperation and quasi-species cooperation, which is more of where that term came about. But yeah, it is one of those cases where I think the same word means different things in English, and then to, you know, different subfields. And so I think that's a great clarification to make.
Yeah, and I think sort of a mutualism perspective would certainly get you less pushback from folks, because I saw you using the term group selection. Now I'm a proponent of the group selection perspective, but a lot of people are not. Yeah. And so that's a trigger term in the field of evolution for a lot of folks.
Definitely. Yeah, and I think the group selection on quasi-species has been also a really, really contentious area of viral evolution as well, for good reason, I think. So yeah, definitely a tricky area.
But you actually may have I mean, you may actually have a really an interesting and potentially appropriate system for looking at that, but it's just, you know, whether it's an actual cooperation or not, but it's certainly it's fascinating. Thank you.
Well, thank you. Yeah. Great. All right. Well, thank you all again so much. It was really